The overall goal of this assay is to evaluate the major modes of clonogenic cell deaths induced by ionizing radiation based on the characteristic morphologies of nuclei stained with DAPI.
This method can help answer key questions in the field of radiation biology. The main advantage of this rapid and cost-efficient technique is that the major modes of clonogenic cell death induced by ionizing radiation can be evaluated simultaneously. Generally, individuals new to radiation biology will struggle to evaluate radiation-induced clonogenic cell death because different assays are required to evaluate each mode of cell death. We first had the idea for this method when we performed large-scale screening of radiation-induced clonogenic cell death using multiple cell lines and radiation doses. Although this method can provide insight into radiation biology, it can also be applied to other systems, such as chemotherapy for cancer. Demonstrating the procedure will be our collaborator Atsushi Shibata.
Start by sterilizing a scalpel with paper towels moistened with 70% ethanol. Then, use this scalpel to place a cover slip in a 35mm cell culture dish. To the culture dish with the cover slip, add 1 milliliter of culture medium. Now, gently hold the center of the cover slip with sterile